Okay, so I thought Chris and I were friends. Being scheduled at the end of such an impressive day of talks, I'm beginning to doubt that. And then, to add insult to the injury, if you like, I have to follow Scotland's answer to Miranda Hart as well. I mean, there is no hope. So now for something completely different then. Fortunately for me, what I have got to tell you today is a really inspirational, good news story. I've been working on this disease, chronic lymphocytic leukemia, for 20 years of my increasingly long life. And I can honestly say to you that the world has changed for CLL patients, absolutely beyond recognition. Even 5 years ago, if someone had said the C word to me for CLL, cure, I would have said, you're completely balmy. But I actually can see a day when I'm going to be out of a job, really and truly. I'll certainly be thinking about something else to do with myself other than just CLL research. So if anyone's got any ideas how I might pay my mortgage, talk to me after. But seriously, really, honestly and truly, the world of CLL therapeutics has changed beyond recognition. And what I'm going to try and convince you of this afternoon in the next 10 minutes or so is how that has come about. So, the outline of my talk, I'm just going to give you the current view of CLL. What do we understand by this word or this term, chronic lymphocytic leukemia? I'm going to try and convince you that this tumour is absolutely dependent on where it lives, the microenvironment in which these cancer cells live. And then I'm going to talk to you a little bit about how new drugs have come along that have allowed us to transform the knowledge that we've gained about the biology of this disease to target that microenvironment in which these cells live in order to kill them and eradicate them. So we're going to talk a little bit about new therapies. And then, I think, just to put the icing on the cake for me, apparently I'm going to end up doing a double act with Professor Bunce. Goodness knows how that is going to work out. Right, so the biology of this disease. It's an incredibly variable disease clinically. Some patients have a normal age-adjusted life expectancy, so they really do not die of their disease at all, whereas other patients have an incredibly aggressive form of the disease, which really should not be called a chronic disease at all. The survival rates for those patients are around 2 years, which is about as bad as any acute leukemia that we have. So, and unlike, say, AML, which Parish and Kirsty work on, when you look down the microscope at these cells, you cannot tell that there's any difference between one patient sample and another. They all look like mature-looking, fairly indolent, benign type B cells, but in fact they behave massively differently. So what we do understand now is the accumulation of these leukemic cells is partly due to an inherent resistance of these cells to die. They're reluctant to give up the ghost, if you like. But we also now know, and this is the critical piece of information that you need to take away from today, that part of the reason why these cells are reluctant to die is not something that's about the cancer cell itself, but about the environment in which those cells live. They get signals from that environment that support them, that help them to stay alive and help them to grow and become a clinical problem. The other thing to say is that when I first started working on this disease, we used to term CLL as a lymphoaccumulative disease, implying really that it was very benign and there wasn't much in the way of proliferation going on, not a lot of cell division. But in fact, we now know that's completely not true. About 1% of the tumour burden, the number of cells that each patient carries around with them, divides every single day. That's a heck of a lot of cell division. So that's changed our view of this disease. I often describe CLL as a liquid disease with a solid tissue problem. And the reason why I say that is the residual disease after we treat it is always found in the solid tissues. So in the bone marrow or the lymphoid tissues in the lymph nodes, not in the peripheral blood. We're very, very good at clearing out that peripheral blood compartment. And like I say, the therapies that we've got are much less effective at targeting those cells in the solid tissues. And we now know that those tissues are the very sites of these extra signals that these tumour cells get, and that makes these cells much less responsive to those drugs. So one of the key questions that we wanted to address, and have been addressing for certainly the last 15 years of my career, is what are those signals, and then can we block them? Because if we want to elicit cures or at least stabilize the condition for patients, we need to be able to target those cells in those microenvironmental niches that they hide out in. So there are 3 different types of microenvironment that CLL cells live and grow in. The first is probably the most obvious one, really. These B cells originate from the bone marrow, and they tend to accumulate in that bone marrow environment. But there's also this microenvironment called the lymphoid tissue, and we now understand that that lymphoid tissue is really where all the action takes place. The cells divide and grow there in a way that they simply don't anywhere else in the patient's body. And then there's this third one in the middle called the vascular niche. So, if you like, where we might derive the term leukemia, the tumour in the blood, okay? And for years and years, people, although we sampled patients' tumours from their blood, we always imagined that the bloodstream was in fact just a passive transit zone to much more interesting places for the cells to go to. But in fact, it turns out that that vascular niche is probably the critical determinant as to the clinical outcome of the disease. And I'll try and explain that to you in the next slide. So, there's interplay between these microenvironments. There's 3 of them, and the cells whizzing around in your bloodstream. And the ability of that cell to escape the bloodstream and go back to this place where they get those extra signals and divide is absolutely key to determining the clinical course of the disease. We never appreciated that until very, very recently. But we have known over the last 3 or 4 years that actually, like I said to you, all the proliferation, and this is the only science slide I've got today, I promise you, that virtually all of the proliferation, all of the cell division goes on in those lymphoid compartments. And as Peter suggested to you this morning, and Christine alluded to just now, and actually Parish mentioned as well, you know, it's cell division that actually drives a term that we call clonal evolution. So the tumour gets changing over time. So the cells that we treat one day aren't actually the same as the cells that we retreat with drugs the next time the patient needs therapy. So there are key questions that my team and lots of other teams around the world, it's important to understand that this is a global fight that we're in, and the LLR is a really important component of that in the UK, and we contribute way above, we fight way above our weight, if you like, in global terms, and you should all be really proud of that. But I think one of the key questions that we've all been asking in this field are, can we establish more representative models that allow us to determine just how cells escape the blood vessels and go back to these tissues, so that then we can understand the molecules, the proteins on the surface of these cells that actually drive that process. And then, by definition, once we know what they are, we can make drugs to inhibit them. So, what's happened? Where's she gone? Okay, Maisie's disappeared. Never mind. Okay, I was going to show you our model system, but she's disappeared. But essentially, I dreamt this up one evening, because I think about pretty much nothing else other than CLL, as my better half will testify, I'm sure. But this picture is even more sort of esoteric than it was going to be. without the other picture there, but this is the inside of a blood vessel, a pseudo blood vessel, one I dreamt up, that we've got it running in the lab now. And these dots here, whoop, I nearly did what you did, Chris. Not quite, though. These are CLL cells. So we've got these cells whizzing around now in these blood vessels, and we can basically look to see how those cells interact with endothelium, the cells that line your blood vessels. This is an electron micrograph of the other side of one of those blood vessels, just to show you that these cells really do escape these blood vessels that we've made in the lab. So it's really very exciting. So just to go back to chemo and why we need to change our view, chemotherapy, as Christine showed you, really works like this. It's the nuclear bomb approach to treating cancer. Okay? And almost by definition, therefore, it's not targeted. Now, this next picture, if it comes up, this is not a selfie, okay? I want you all to be absolutely aware of that. His ears are slightly bigger than mine. Anyway, we have started to think a lot more smartly about how to devise treatments for cancers and leukemias in particular. As Chris said to you this morning, we're really in the vanguard as leukemia researchers and clinicians in devising new therapies. A lot of the advances that are being made in cancer research in general start their lives in leukemia research laboratories. So, this is the first smart therapy that came along, really, monoclonal antibodies. We can argue about whether there were other more targeted drugs before these, but essentially these changed the world as far as I'm concerned. Monoclonal antibodies, they targeted particular proteins on the surface of cells and allowed us to target our therapies to a particular subclass of cells, usually cells, well, the cells that are the leukemia. But they're not specific. We need to remember that. They're targeted, yes, but in this case, this is targeting a B cell, and we all have normal B cells as well as malignant B cells in the case of CLL, and those cells get affected too. So they're not truly targeted in that sense. And this is just a slide. I don't need you to remember any of this. There won't be an exam. But these are all the new monoclonal antibodies that I could think of off the top of my head that are in clinical development. So you can see that the world is vastly changing now in terms of devising these new targeted therapies. Just to reiterate for you, in terms of developing new therapies, why do we need them? Well, because in the case of CLL and other forms of leukemia, in the peripheral blood, we can view that almost as if it's like a desert in terms of supporting microenvironment. And we can use the conventional drugs we have very effectively to target those cells in this relatively unsupportive environment called the peripheral blood. However, even monoclonal antibodies don't work very well in our oasis that we might call the lymphoid niche. Okay? This is where the cells grow, and this is where we really do need to focus our attention and target therapies. So, new treatments for CLL. This is the good news bit. There are lots. Okay? And I'm not going to talk about all of them, you'll be pleased to know, and so will Chris be, because he's going to wind me up any second now. But essentially, I'm going to talk to you about just about two. I'm going to talk to you a little bit about chimeric antigen receptor T cells. You've heard about that this morning as well. So those of you who are listening, this will not be a new phenomenon to you. And then I'm going to talk to you very briefly at the end about the transforming new class of agents that have already changed the landscape of CLL, these B-cell receptor inhibitors. So, CAR T cells. These are T cells we take from patients, and we can modify those T cells to make them recognize the patient's own tumor. Okay? So, really smart idea. And the idea is that they They leave normal tissue untouched because the T cell doesn't get activated, because it doesn't receive its second signal. But when it sees the tumour cell, the T cell becomes activated and it kills the tumour cell very effectively. And we know this works. I think Carl June told us it does. So it must be right. And it's in the New England Journal of Medicine. But what I would say to you, although these drugs are, these new cellular drugs, if you like, are incredibly powerful tools, there are no tumour-specific antigens as yet. So they still don't target specifically the tumour cell. There are a couple of other little problems with these type of drugs as well. The T cells are derived from the patient, and the patient's T cells have already worked incredibly hard to try and fight off the patient's leukemia before they've even got to become changed by these really fancied-down scientific processes that we do out of the patient. So the T cells can become exhausted. They can give up the ghost before the tumour is eradicated. So we've still got work to do. And I would question, in the long run, whether these are going to be better than monoclonal antibody therapies that are already being pushed forward into clinical development. Fortunately for us, we might not need CAR T cells in CLL because we've devised new ways. This is all based on the biology that we've understood about this disease for targeting the B-cell receptor signal. So this is something that is nothing to do with genetics, okay? Peter talked to you this morning, and it's undeniable that our understanding of the genetics of human cancers in general and leukemia specifically are transforming our knowledge of these diseases. But not everything comes down to genetics, because this is just about the way that the cells normally would behave. They see an antigen, because that's what a B cell does, and in response to that antigen, they divide. That's what all of us are doing when we face infection, whenever we come across it. So this is normal cellular process. It's not a genetic aberration. Okay? But what people have done is realize that these antigens are chronically stimulating CLL cells, and this is promoting growth. So this drug is the first in class, ibrutinib, but there are lots more coming along in its train. And as Parrish says, you know, these drugs have to be affordable. And I think the main way that we'll make these drugs affordable is to create competition in the marketplace. Okay? So we want more pharmaceutical companies to lack the intelligence and insight to develop new agents and new therapeutic strategies. We want lots of B-cell receptor antagonists to come along so that we can drive down the price of these drugs for patients. Okay? But this ibrutinib is a wonder drug. The clinical trials data, which I'm just going to go over with you for 1 second, are quite startling. Remember, this is all data generated in relapsed or refractory patients. So these are patients who've reached the end of the line as far as CLL therapies are concerned. Okay? So this drug, it blocks this molecule here, Bruton's tyrosine kinase, but you simply do not need to know that. But essentially, it stops the cell getting activated and it stops it dividing. It's already licensed in the US, and it probably will be licensed in the UK this year. 75% of patients who got this drug had progression-free survival for, well, over 2 years. And 83% of those patients showed overall survival at the same time point. So this is, remember, these patients would be termed almost untreatable without this agent. So it's a remarkable result. The most important thing to remember is this drug is really effective in high-risk patients. So we really didn't have any options for these patients beyond multifaceted chemotherapy with a monoclonal antibody. So it really is changing the world. Okay. The interesting thing, and this is something that we've made a small contribution to in Cardiff, is the mechanism of action of these drugs is really primarily to stop those cells escaping the blood vessels and going back to the lymph nodes. So a completely different therapeutic strategy to the ones that we've had previously. And you could argue, with a lot of justification, although this hasn't been tested yet, that we may actually be able to kill off all of these tumour cells using chemotherapy because they get trapped in the peripheral blood, and we know that chemo works really well in that environment. So I'm going to shut up. But essentially, we've established this model, and we've learned how to block the molecules that are key to driving that escape from the blood vessels. And that is really changing the world for CLL patients. Okay, so new therapies have emerged based on the biology of the disease. Patients with refractory disease and high-risk features really have hope for the first time. These inhibitors have a completely different way of working. And really importantly, and this is where Chris is going to come and join me, I think, these drugs are likely to, in fact, we know that they're going to have really exciting activity, not just in CLL, but in other B-cell malignancies. So I'll stop there.